Okay, so good afternoon, everybody. I hope you enjoyed the lunch. So this afternoon, first lecture is about ecosystem health indicator and how we derive indicator from ocean color observations. So I'll start with a nice animation provided by Trevor. Actually, it's provided by NASA. So on this animation, you can see the ocean. You can see North America, Africa, Europe. You can see the sea ice changing over time, the Hudson Bay. For some of you, it's of interest. And you can see changes in the concentration of chlorophyll and changes in phytoplankton. So where you have red color is where you have high chlorophyll concentration, and blue color is where you have a very low chlorophyll concentration. And I think this image is very interesting because you also see on the continent the changes in the vegetation. And so you also, you see both on the ocean and on the continent changes in the seasonality of the phytoplankton. And you also see variations. Here it's the equatorial Pacific. And you have a very strong bloom just along the equator. And I think my talk is on Thursday. We'll discuss more in details about the influence of El Nino climate phenomenon in that particular region and how it can enhance phytoplankton concentrations. So you've seen now on this image how we can see the seasonality changing, how we can see phytoplankton chlorophyll concentration changing. So this was really to show you partly the Earth system component. And so I would say when we talk about climate change, climate drivers, looking at impacts and responses, we really have two systems that are interacting. So the Earth system, and I would say the human system, so us, our society. So we are a very strong driver into changes in the environment. So drivers because of our socio-economic development, a driver because of population development, population growth, and industrial revolution. Okay, we're starting emitting CO2 in the atmosphere, so we are actually influencing the Earth system. We are changing concentration of CO2. We are changing some of the climate processes. And those climate processes then, lead to changes in climate. So we can see them on land in terms of changes in temperature, in the ocean, sea level rise, changes in precipitation, more extreme events. In the ocean, influence of changes in climate. So we will see this in terms of oxygen concentration changes, nutrients might change in the ocean. Of course, the temperature of the ocean is going to change. The acidity of the ocean, the pH of the ocean is changing. To give you some examples of the changes in the ocean. And of course, this will have an impact on the ecosystems. So they will respond to these changes. This will have also an impact on the water resources. And if you have changes in the ecosystem, then this will go back to the human system. So this will have an influence on food security, food supply, for instance, fisheries coming from the ocean, and water resources. So it can also have an impact on human health if you have some development of more pathogens, let's say, because you have changes in environmental conditions. And then again, this goes back to socio-economic development. So if we cannot have food security or we cannot ensure good human health, this will influence settlements, and this will influence the entire society. So you also have here, it's not just a one-way loop system, it's also, you also have feedback mechanism. So the ecosystem respond to changes in climate. And we will see during the week actually how phytoplankton can itself have an influence on the climate. So phytoplankton plays a huge role in the carbon cycle. So we'll have a lecture on this. It also had an influence on the ocean heat flux. Shuba will give you a lecture on this. Some phytoplankton species can also play an influence on the climate. For instance, dimethyl, Phyocystis species, they will produce dimethyl sulfoniopropanate, so this will have an influence on the clouds formation in the sky. So this will have an influence again on the climate. And okay, so we will see some examples of this during the week. So why do I go so much about this? Well, because this course is also to place ocean color data in a context of climate studies. So I wanted to give you a little bit of an introduction, a bit of context related to this. Should have mentioned that this document, this schematic framework is from the IPCC report. So IPCC is the International Panel for Climate Change. So it's an old report. I think it's the assessment report number 4 that was published in 2007. In the meantime, there has been assessment report number 5 in 2014, 2015. And now they're looking for assessment report number 6. So as I mentioned, we have drivers, so environmental and social development. So actually an indicator of this is actually changes in the world population. So we see, I think nowadays we're reaching about 7.5 billion people on the planet. By 2050, the target, no, it's not really a target, but the projection are for 9.8 billion. And so actually this increase, this growth in the population actually is having an influence on our global emissions. We need energy. So so this has been driving in the past, so the increase in atmospheric CO2. So again, so this is another type of indicator. It's not an ecological indicator, but it's an indicator when we measure the concentration of CO2 in the atmosphere. So we know how our global emission changing. And so as we've just described it in the previous slide, so these changes in the climate processes and the climate itself will have an influence on the pelagic ecosystem. And so now I'm going to describe to you ecological indicators. So related to the functioning, the structure and the composition of the ecosystem, and how can we use them as resource management tools to support a comprehensive assessment of the ecosystem state. So indicators, the term itself, they've been used in many, in quite a broad context, so they can be used, of course, for to monitor and to give you some information about the environment. But we also have, so we have environmental indicators. You might have economic indicators. You have a very broad range of indicators. So these indicators can be very discipline specific, but they can also be used to link different disciplines. So for instance, you can have socio-economic indicators, or you can have environmental economic indicator. You even have a journal that is dedicated to this. Or you can have indicators that encompass several disciplines and to give you a more holistic assessment of your system, and then we are talking maybe about sustainability indicators. Okay, so I'll give you some examples of ecological indicators. So these are more general, not specific to ocean color so far. So it's actually the subject is treated in great details in the publication of Rice 2003. Where he provides a number of categories of, he describes a number of categories of indicators. So they can be indicator species. So it's a lot of text. The first 3 slides are like this, and then it's much less text. But I thought it was useful for you to have it. So indicator species, so looking at biomass and mortality rate, diversity indices, so how many species are present in a system or in a collection or in a sample. You can also have indicators based on ordinal methods. So looking at more statistical multivariate analysis, so for instance PCA or cluster analysis. Once you have a number of indicators, they might be aggregated in order to give you more information about the system. And you can have indicators that represent, that give you some information on emergent properties of the ecosystem. So, for instance, looking at resilience to species invasion or losses, stability over perturbation, parameterization of biomasses and flow rates, and transfer efficiencies and distribution of biomass among different trophic levels. So these are some possible categories of indicators. It's not at all exhaustive. Okay, so the selection of indicator will be compromised of many factors, but it's usually selected and optimized for the intended purpose. So what is the goal? What are you trying to measure? So you will get a specific indicator related to this. But also the selection of this indicator is often limited or constrained by the availability of observations that you have. And is often also limited to a specific stream of observations. So it's not so easy sometimes to estimate indicators from different streams of observation and then compare them. I'll give you some example on how we've done this through maybe three case studies during the talk. So again, so the next two slides, a lot of text, again from Rice. So he did a very extensive review on this topic of ecological indicators. And he proposed a number of criteria for assessing and ranking indicators. So I think it's quite useful to look at the list for several reasons. So first, is this indicator going to be meaningful? Can it be understood by the correct audience that you're looking at, whether it's scientists, whether it's decision makers, policy makers, whether it's the general public. You might want to use different indicators so that they can be readily understood. The measurements in order to collect information about this indicator. So what needs to be done to provide the data? Can it be done on a regular basis? Then we might be interested to know information about the accuracy and precision of the indicator. So I'll actually give you some example in some slides about uncertainty estimates using ocean color observation and specifically looking at accuracy and precision. The representativeness of this indicator. So can the dynamic of the indicator be taken to reflect more than the dynamic of the specific times and places where the data were collected? So can you make some extrapolations? Can you record seasonal variations in the properties that you are monitoring? Can you know a bit more on the geographical variations as well? So availability of historic data. So is there a frame of reference in which to build an understanding of the performance of that indicator? Okay, and the second long slide is about specificity of the indicator. If it changes value, how indicative is it of a change of a particular forcing factor? The ability to set reference points for that particular indicator. Can we detect regime shift or tipping points from this indicator that we will be measuring? So sensitivity, can the indicator be trusted to inform users fully if the environment really is undergoing change in properties? The responsiveness, will the indicator give feedback on ecosystem change on timescales that are useful in management decision making? And then we go to legal consideration. Is the indicator something for which a legal requirement to report exists? And a theoretical basis, is there a sound basis in the ecological theory for why the indicator should be reliably informative about the effects of particular forcers or forcing processes? So here are some just from this list. And so now we'll move on to the indicators that we can derive or estimate using ocean color observations. And we'll see actually whether some of these criteria apply to these indicators. So learning objective is to establish and assess the value of remotely sensed imagery for the development of ecological indicators in the pelagic zone. And I will give you some examples of applications in different regions, so around the UK, in North Atlantic, Nova Scotia shelf, Atlantic Ocean, and global scale examples. So a little bit of context. So more context, actually. There is a global consensus that management should have an ecosystem basis, and the integrity of the ecosystem should not be compromised. So ideally, we would want to maintain ecosystem health attributes such as vigor, structure, resilience, and resistance of that ecosystem. However, such concepts such as vigor and resilience of the ecosystems they tend to be quite subjective, and they are difficult to quantify. So instead, actually Trevor and Shuba and other authors, but they were very strong into this, develop a suite of ecological indicators as an aid to ecosystem-based management. So these indicators are objective metrics for the pelagic ecosystem, and they can be applied serially in operational mode to detect changes that may occur in response to environmental perturbation. So ideal characteristics of pelagic indicators, so they should represent well understood and widely accepted ecosystem property. They may be quantifiable unambiguously in standard units. They can be measured rapidly and at low incremental cost. So their repeat frequency may be compatible with intrinsic timescale of properties under study. They are measurable at a variety of scales, and possibly they can allow us to create long, maybe multi-decadal time series. And so you've seen since this morning that nowadays we are nearly reaching 2 decades of observations. So we are creating a long time series of ocean color observations. And so actually remote sensing is actually meeting the criteria for operational metrics in terms of speed, resolution, repeat frequency, and cost effectiveness. Autotrophic biomass, so the biomass of phytoplankton, so the base of the marine food chain, is a very important ecosystem property. So fields of primary production, so are you all familiar with primary production? Yes, so it's a rate at which carbon organic carbon is being produced. So fields of primary production can be generated. SST and chlorophyll can be obtained at the same resolution based on different sensors. And we can construct time series that will allow us to look at the seasonal dynamics and to quantify this dynamic in an objective way using different metrics that I will present. And we can perform interannual comparisons. So here are just some examples. So it's UK shelf seas, and we see chlorophyll concentration. And here on the right-hand side, sea surface temperatures, both obtained at the same resolution for the same period from remote sensing. So these represent fundamental properties of the ecosystem. why they do so? Well, chlorophyll, it gives us an idea of autotrophic biomass, the base of the marine food chain, and sea surface temperatures, and it's a very important properties for the dynamic of this phytoplankton. So here are a list of ecosystem indicators that can be estimated from ocean color remote sensing. So the list was proposed first by Trevor and Shuba. So you can find the list in that particular publication. In 2009, it was first put up in 2008, but maybe this list is a bit more exhaustive. And so you see have a number of indicators, and we will go through them, and I will give some example of not all of these indicators, but quite a few in the next few slides. Some of these indicators relate to the timing of events, so the phenology of phytoplankton, and some are more based on related to absolute changes. So maybe annual phytoplankton production, 19th percentile of chlorophyll. I think maybe tomorrow you will hear about the initial slope of light saturation curve, assimilation number, and you will hear more about the carbon indicators as well in the next few days. And I will also briefly mention, and we've mentioned this morning about phytoplankton functional types, and the delineation of biogeochemical provinces. I will touch upon it in some of the slides. And here you have the dimension. So we see that some of the phenology indicators are more in the time dimension. Well, maybe amplitude is in milligrams of chlorophyll per cubic meters. So you see the different units. Okay, primary production. Okay. And some are dimensionless, and some don't have a dimension. Okay, so from remote sensing, we have daily images, or sometimes even higher frequency, higher resolution, time resolution. But let's say we have daily images, so we can create composites of several days, maybe 5-day composites. So we take the average over 5 days, or 7-day composite takes the average over 7 days. Can also create a monthly composite. So we do this in order sometimes to reduce gaps because we have cloud cover. So by making composite, maybe we get better coverage. And we can create a time series at, let's say, monthly resolution over now 18 years. And if we look over 1 year, we have here the schematic representation of the changes in biomass, changes in chlorophyll concentration over time. So from this signal here, we can estimate some indicators of fundamental ecosystem properties. So for instance, here is a group of them. It's not exhaustive. We've seen in the previous slide there are more, but some can be timing of the spring maximum. So when does it peak? The amplitude of this signal, the annual phytoplankton production, and get some information on phytoplankton functional types depending on, for instance, chlorophyll concentration in the water. And we can also aggregate this group of indicators in order to help us to provide a more holistic, more comprehensive, to get a more comprehensive knowledge or of the state of this ecosystem, and then use this information for ecosystem-based management. So here we're back with this schematic representation of the changes in chlorophyll over time. So this is, let's say, an annual cycle, so it would be over 1 year, and we see the changes in the seasonality. So maybe if we are in the northern hemisphere, maybe in winter we tend to have a lower chlorophyll concentration, and as we go towards spring we get an increase in this phytoplankton concentration, maybe reach a maximum and then decrease. So this would be a case where we have only one phytoplankton bloom or one phytoplankton growing period in the year. And so we can estimate some of these indicators. So we've just discussed it, the amplitude, the timing of peak. We can look at annual mean chlorophyll, annual mean primary production. We can also estimate the timing of initiation, so when does the phytoplankton growing period is starting. timing of termination, when does it finishes, and the duration of that growing period. I will go in details about phenology algorithm in the next slide, because this is what I've worked a lot on. And you will get more details about other indicators in the coming days when my colleagues introduce their topic. Any, or actually all of these indicators may vary between years, so this is interannual variability. And this can happen, of course, at any pixels of the region of interest. You will get variation in this cycle. And so some of these indicators can be, sometimes we can look at them in terms of absolute and relative indicators. So what I call absolute indicators, it's when they are based on absolute changes. So they will be expressed in units of, for instance, milligrams of chlorophyll per cubic meters. Or if we look at primary production, changes in grams of carbon per meter square per year. It's a rate of change, so we have a time unit. And so these indicators would be sensitive to errors in the absolute values of chlorophyll, which may arise from during the calibration or the development of the retrieval algorithm for different sensors. And then we might look at indicators that are based on relative patterns to avoid the sensitivity to these absolute to errors in the absolute values of chlorophyll. So this might be indicators of looking at changes in timing of this seasonality. And so these indicators are expressed in units of times, for instance, number of weeks, months, a day. And so they will not be so much influenced by uncertainty in the actual absolute values of chlorophyll. Maybe you, in some region, depending on the algorithm that you are using, you might be slightly overestimating the chlorophyll or underestimating the chlorophyll. But because we are looking at timing of events, which will be based generally on a relative threshold, then you're not so bound by these errors in the absolute values of chlorophyll. So it's just a way to look at those indicators. So I'll go on with phytoplankton phenology. And here on this map, we show you the timing of peak of phytoplankton chlorophyll for one bloom, the main bloom during the year. And so we see this at global scale. And so actually the color scale on this map is actually showing you a timing of an event. So if we look, for instance, in the North Atlantic, because it's an area that maybe I know a little bit better, maybe we have initiation around here at the lower latitude, maybe around February, March. And as we move towards the higher latitude, we see that maybe the timing is more towards the months of April, May, and as we move towards very high latitude, maybe more June, actually, a blue color. Well, in the southern hemisphere, so it's opposite period, so we would have the spring bloom more during the period of maybe October, November, towards December and January, as we move towards the pole, towards the Antarctic. So this algorithm has been developed. It's simple. Here we show the timing of peak, but we can also show timing of initiation, for instance. And in order to estimate this, it's quite simple. We take a relative threshold. For instance, over a very long time series of 18 years, we might take the median chlorophyll concentration and estimate median plus 5% or median plus 10%. And this gives us a threshold. And once the chlorophyll concentration during the year goes above the threshold, then we say, okay, this is a time when the phytoplankton growing period started. And when it falls below the threshold, it's when the phytoplankton growing period terminated. the number of weeks in between the termination and the initiation and termination is the duration of the phytoplankton growth. So this was for the global ocean. We've also looked at regional algorithms for the Red Sea. So actually you will have the practical on Wednesday will be related to that region. So looking both at chlorophyll concentration but also phenology. And in the northern, is it northern Red Sea? Actually no. So in the Red Sea, in some regions of the Red Sea, let's say around here, you might find that you have 2 growing periods. One during what they call the winter periods. So bear in mind that here it starts October, November, December, and so on until August, September, and the summer period. So you might have the larger bloom during the winter period and a smaller bloom during the summer period. So we simply fitted a threshold that would allow us to compare both the winter and summer. So we use the same threshold. And again, we could get some maps. So this is the Red Sea. So the Red Sea is here for the characterizing the winter peak. So it's again showing the timing of the peak here and the summer peak. So in the northern Red Sea, there is only a winter peak. There is no summer peaks. While in the southern Red Sea, you get both winter and summer. And actually, with the advance of the Ocean Color Climate Change Initiative, the fact that we had several sensors recording information, we had better coverage. And also the use of a new atmospheric correction algorithm called Polymer allowed for the first time to actually see through some very high-level clouds and get better coverage even during sandstorms or light sandstorms. So we had a much better coverage, and it was the first time that we could characterize the summer phytoplankton bloom in the southern Red Sea and the whole northern Arabian Sea with the CCI data. Okay, so this is for these algorithms. So a little bit more information on one of the recent approach that I developed, so with the CCI data. So here you have a data stream from OCCCI, so satellite observation. You might create bins at 1 degree bins and make 5-day composites. So the idea to do this is simply because if you have a larger box, then you put in more data. So maybe again you get better coverage or fewer gaps in your time series. Because actually you can imagine that if you have a gap somewhere, you might be missing the timing of peak or the timing of initiation. So you want actually gap-free time series. So that's why also you make temporal composites. For the very high latitude, where actually the satellite cannot record information during the winter because of high solar zenith angles, and I used NOBM data, so it's a NASA Ocean Biogeochemical model. So only for that period, instead of having a gap, then I simply filled. So why this particular model? But simply because this model actually assimilates chlorophyll during the entire time series that they have. So assimilates satellite chlorophyll into the model. Stefano will explain to you a bit more about data assimilation and how to assimilate chlorophyll. And so by doing this, it was the concentration were much more easily comparable. So I could simply fill in. So where it shows in red, it's at high latitude. I think maybe it's 65 degrees north here. So I could simply fill. Okay, so we have a continuous time series. And I also use, so the climate change initiative from ESA was for ocean color, but also other essential climate variable. Sea surface temperature is one of them, together with sea level as well. I will mention about it during the week. And so I simply defined cooling and warming periods for each year. And simply then I search for timing of phytoplankton growth during both the cooling and the warming period. So in this case, we see some years we have, so the main peak is usually during the warming period, but sometimes you also get secondary bloom during the cooling period of lower amplitude, but a secondary bloom. And so using that method, then simply I used a derivative of a cumulative sum of anomalies to smooth the signal, and then I could retrieve phenological indices. And what do we get is then timing of chlorophyll peak during 2 periods. Here is June, July, August, September, October, November, and a peak timing during December, January, February, March, April, May. Okay. And if I want to combine the two maps, okay, well, simply if I want to look at the main chlorophyll peak, so I take the one that is the strongest at every pixel in the ocean, then I get just a map with this main chlorophyll peak, rather than having a map with 2 peaks. Are you able to follow, or have I lost everybody? Yes, okay. Okay, and so other metrics that we can derive is not just the timing, but you can also get an estimation on the probability to have 2 chlorophyll peaks per year. So you will find that in some regions, generally lower latitude, tropics and subtropics, probability to have 2 peaks is 0. So that means that in this region, you usually get only 1 growing period. Sometimes it would be very low increase in chlorophyll, but you can have a little bit of an increase. So you would have just one growing period. And as you go towards higher latitude, you see that it gets darker colors. So it means that when you get very dark blue, it's 1. So it's every year you have 2 chlorophyll peaks. And where it's lighter blue, it's 50% chance to have 2 peaks. So some years you have 2 peaks, and some years you have only 1 peak. So, okay, it might seem a little bit strange, but actually in a study, modeling study done by Trevor and colleagues, very simple model forced by light availability. They compared at 45 degree north and 65 degree north around this region, and they actually found the same results just based on a simple model of phytoplankton forced by light availability. And some years they would have 1 peak, and some years they have 2 peaks, and again just 1 peak. While going towards higher latitude, so maybe more around here, they had systematically just 1 peak. So it was quite interesting to compare the two, and actually to validate some of this model observation with satellite observations. And what you can do also, it's simply to estimate the probability that the main chlorophyll peak will occur during which months. So either during June, July, August, September, October, November. So generally in the southern hemisphere, this is kind of the spring, summer period. And in the northern hemisphere, generally the main peak will occur between December and May. So this is where you get a probability of 1. But you also find some regions, also in the southern hemisphere, where generally the main peak, I'm not saying there are no 2 peaks, some regions might have 2 peaks, but where the main peak will be during that period, and around Madagascar as well. It's quite peculiar there. Yeah. Yes. Yeah. Yeah, I think the 65 is somewhere there where it's white again. So yeah, here some parts you systematically have 2 peaks, or actually it's estimating 2 peaks. But here, right there at around 65, it's just one peak. And here below, maybe 45, maybe somewhere. It should probably be somewhere in the 50%. Yeah. Actually, I made a mistake when I said this. I don't expect higher latitudes to have two peaks. It's usually more one peak, actually, if you go really toward a much higher latitude. If you go in between, maybe to have two peaks is simply because you get larger variation in your in those regions, maybe larger variation in your light, in your irradiance availability. So that could be the reason, if I'm correct, yeah. Yeah. Okay, so this was for phenology, was an example of indicator. So I've shown you some a little bit more details about this. I won't go into more details in the next slide that I'm going to show, but I will show you a couple of slides with examples of other indicators. So tomorrow you will have an entire lecture on primary production and modeling primary production. So I just put it here because it's a very key indicator. And so here is just to show you estimates of primary production. So this is a global scale. They vary between 45 to 50 petagrams of carbon each year. So this represents actually 40%, about 40% of the global primary production, including the terrestrial system. And so probably some of you already know, but petagrams of carbon per year is equivalent to 1 gigaton, 1 billion ton, and here are in it's 10 to the power of 15 gram. So it's enormous, basically. And you find you get higher primary production at higher latitudes and lower primary production in the gyres. Okay, another indicator that I mentioned is 19th percentile of chlorophyll. So if you take a time series of chlorophyll over several years, maybe you estimate, so the 19th percentile is when the chlorophyll concentration goes about 95% well, if you take your time series, you take the 90% of your chlorophyll concentration, and anything that goes above that 90%, if you rank your chlorophyll, then this is when you exceed that level. So you can, maybe it's an indicator of when you get eutrophication or very high chlorophyll concentration, maybe something is happening in your system. So here is for the UK shelf seas, and so this is just maybe annual concentration. And here is your 19th percentile, so it tells you along the coast you see that the 19th percentile is a much higher concentration. And here is just an example for at one specific month or one specific day when in red when the chlorophyll concentration exceeded the 19th percentile in that place. And in green is when it was below that threshold. Okay, carbon-related indicators, so there are quite a few, and I think you will hear more about them with Bob, who will introduce you the ocean carbon cycle. So you can have estimates of phytoplankton carbon. So here is an example from one of Shuba's paper based on chlorophyll concentration, total particulate carbon as well from chlorophyll concentration, and a carbon to chlorophyll ratio based on phytoplankton carbon and chlorophyll concentration. Then you can derive maps. So you will hear more about the development of this algorithm, I think, with Bob. Phytoplankton size structure, so we mentioned it this morning. So here is maybe an example from Hirata et al. Based on different chlorophyll thresholds. So in some regions where chlorophyll was above 0.95 milligrams of chlorophyll per cubic meter. So, okay, generally it was micro phytoplankton. And so you see here on the map, so this is the UK. And in red would be places where you have more micro phytoplankton. Green is nano phytoplankton based on those thresholds. And blue is where you would have pico phytoplankton. I think Trevor showed this slide this morning, the algorithm of Brewin et al. So it's been refined again in 2014 and maybe again recently, also based on concentration of chlorophyll. So it's a 3-size component algorithm, again for pico, nano, and micro plankton. So this one has been developed for global oceans. And here it's expressed not in different colors like here. It's a percentage of pico phytoplankton in that region. So generally, this is maybe here in that region 100%. So this is generally at that time in particular, in May 2005, you would expect to have 100% would be of your phytoplankton population would be dominated by pico phytoplankton. And of course, you find very low percentage for nano and 0 for micro. I think although we mentioned this slide this morning, so there is more to phytoplankton than carbon. So they are also a major player in biogeochemical cycles, including, for instance, nitrogen, silicate, iron, and calcium. So here are some examples of phytoplankton functional types. So they play a role in the biogeochemical cycles. So we have silicifiers, so diatoms. They have a very important application for carbon export. They are very large phytoplankton, and they will sink very rapidly to the bottom of the ocean, so they can export a lot of the organic carbon as they sink. Calcifier, so they will play an important role in ocean acidification, as well as the carbon cycle. Again, they tend to be quite heavy phytoplankton cells, so they will sink very rapidly and export a lot of carbon as well. DMS producers, Phyocystis, so I mentioned them earlier. So they produce dimethylsulfoniopropanate that will get released to the atmosphere and will play a role in the formation of cloud condensation nuclei. And so this can have an important feedback on the climate if you produce maybe more of if you more cloud condensation nuclei, then you might get more cloud formation, and you can reflect some of the incident sunlight back towards the atmosphere. Pico phytoplankton, so they play an important role in recycled production. So they are maybe generally found, as we can see on this slide, more in the tropics and more in the subtropics actually. And diazotrophs, so for instance, I think Synechococcus-like cyanobacteria, so will play an important role in the nitrogen cycle. And so here is an example from of a method that has been developed by Alvin et al., based as well on the reflectance of the signal. So you get different, and she's able to map different phytoplankton functional groups, including diatoms, so they are shown in red on those two maps. So it's an example for January and June. Diatoms are shown in red, so actually these pixels tells you that the water are dominated by diatoms. It doesn't exclude that there may be other phytoplankton present, but it's more about dominance, so it would be dominated by diatoms. We see them in the North Atlantic here. Coccolithophores, we find them around here. Phyocystis, so the DMS producers, are estimated in the Southern Ocean. Okay, nanocaryotes, so very small phytoplankton everywhere else, and Synechococcus and Prochlorococcus. They are quite difficult actually to distinguish, but still the algorithm gives some differences, and they are more found in the tropics and subtropics. So this slide has been shown this morning. So it's an algorithm developed by Shuba on the probability of occurrence of diatoms. So Trevor explained this figure earlier on. And so I added just one slide on the relevance of why do we want to detect whether there are diatoms or no diatoms. So this can have an important influence on the food web. So this is an example of a food web structure from a paper by Curie et al. 2008. And well, actually, it was looking at upwelling ecosystem in, I think it's the Benguela upwelling ecosystem. And in years or in times when he had diatoms, then generally he found the larger copepods in his ecosystem, and this would be more favorable for anchovies. And in times when there are no diatoms, so he might find smaller phytoplankton and flagellates. So this would be more favorable for small copepods, and small copepods would be a more favorable food source for sardines. And so this is how sometimes you can have maybe shift between sardines and anchovies in your ecosystem. So this information based on remote sensing is very important to understand trophic interactions. So here is an example on harmful algal blooms. So you will have a practical class on, I think, Thursday morning, if I'm not wrong, with Hayley on the detection of harmful algal blooms. So she will introduce you as well to the development of the, I think, algorithms. Here is an example, it's two examples. It's a bloom here, this is a North Sea. And it's a bloom of Chatonella, which it's a toxic phytoplankton species that killed 350 tons of Norwegian farmed salmon in 1998 due to their presence, their bloom. And here is another example, so you recognize Plymouth somewhere around here. Carenia, that so dinoflagellate, generally I think it generates red tides and is very toxic, and it killed 500,000 Shetland salmon in 2001. And so here is an example from Peter Miller, who is working here at PML, who developed algorithm for harmful algal bloom detections. And in those regions, so he actually maps of harmful algal bloom likelihood. So where it's in red, you have high chances to have harmful algal blooms, and where it's in blue, you have very, very low chances. And what he developed in a project on AlgaRisk, so information on which region are at high risk and where, for instance, the fish farming might be affected. So he gave a simple color. So green is fine, yellow is probably high risk, and red is definitely present. And so this is also used to inform southwest basing area, so around the southwest UK. So this is an example actually where we inform policy and water regulation bodies. And subsequently the population. So a completely different type of indicator. At the bottom of the list there was something about biogeochemical provinces. And so longest 1995 and then 1998. Some of you might have heard of his very famous book and very famous partition. I think he developed it in collaboration with also Shuba and Trevor, partition of the ocean into a number of biogeochemical provinces. So his delineation was static. Simply, it was based on information that he had. I think he included some satellite observation, but it remained very, very static. And more recently, so with the advance of remote sensing and more availability of the data, using both chlorophyll and sea surface temperature and classification method, I think K-mean cluster analysis, Devred et al. So developed a more dynamic partition. So they entered the same number of provinces that they were expecting, here in color, in the cluster analysis. And then they can generate maps at maybe, let's say, monthly resolution, and they can see how the boundary of the different biogeochemical provinces are changing and evolving through time. So this is an example. Okay, so I think this slide has not been shown. So ocean color radiometry, so we can derive, so we have a primary product. What kind of primary product? So I think Trevor mentioned this morning about apparent optical properties, water living regions, reflectance, absorption, yes, okay, it will come back. Inherent optical properties, so absorption, backscattering, and constituent contribution, so specific absorption from water, phytoplankton, seedoms, and non-organic particles, and also aerosol products. So these are examples of primary products. From these, based on those primary products, an auxiliary information, for instance, SST would be an auxiliary information. The power availability of light would be an auxiliary information. We can derive secondary products. Okay, and also in situ measurements to develop a chlorophyll retrieval algorithm. Primary production we can develop using PAR and also sea surface temperature. So you'll hear about it tomorrow. We can derive phytoplankton groups, functional type, phytoplankton size classes, information about particle sizes, POC, DOC, euphotic depths, suspended particulate matter, and phenology. So these are some examples. It's not exhaustive list. And having those secondary products, and we can use them, both the primary and secondary, to address specific scientific issues. And then we can actually address and transfer to a societal benefit area. So transition from research and development to operational oceanography. So for information for the benefit of the society. So where am I going here? So here are some examples on where ocean color application can be used for the benefit of the society. So here it's a schematic from the group on Earth observation. So Sophie will mention about it, I think on Wednesday. And so it's a group, I think it's voluntary, so it gather different institution and organization and different government to develop global Earth observation systems that will take information from different sensors. So satellite sensors, ship and floats and other sensors. And to address and to provide information for the benefit of the society in a number of societal benefit areas. So looking at disaster, health, energy, climate, agriculture, ecosystems, diversity, water, and weather. And actually, recently, I don't know the exact year, but Sophie will tell you. So there has been an addition to this list, which is a Geo Blue Planet initiative. So I think now they should have an extra box here, if I'm not wrong, Sophie.
The whole structure
has changed. Okay, so I'm out of date. I'm sorry, but still okay. And here I've mapped some of the ocean color applications. So in terms of health, harmful algal blooms are very useful. Climate, so phytoplanktons play a huge role in the carbon cycle, play an important role in terms of climate feedback for agriculture. So in terms of fisheries and aquaculture, it's the base of the marine food chain. Ecosystem dynamics, so play a huge role for the marine food web dynamics. Biodiversity, so when we look at phytoplankton functional types or phytoplankton size structures. And this ocean color data, actually, Trevor mentioned it this morning, you get the most information when you can use different data streams. And so here is an ecological framework with a number of plankton indicators. And different observing systems. So the list is not exhaustive, but it's just to give you an idea on some of the indicators that can be estimated from mooring site, ship, from during research cruises, glider or Argo floats, based on an optical sensor, continuous plankton recorder, and satellite observations. And those plankton indicators have been classified into a number of ecosystem and attributes. So that relates to gives us information about the composition of your ecosystem, its structure. So here we have, so in terms of composition, looking at taxonomic diversity. So this is a key example. Structure, so looking at chlorophyll concentration, the carbon to chlorophyll ratio, phenology, and also looking at the functioning of your ecosystem. So here you have changes in primary production, so rate of production of carbon, for instance, and photosynthesis irradiance parameters. So having knowledge on different ecosystem attributes, then you can provide information, so you can aggregate this indicator information to provide information on the state of the ecosystem, and maybe measure the impact of climate, for instance, or different climate phenomena on ocean primary producers. So I will now give you some examples as to maybe why it's quite useful to make sure that you are looking at indicators in that belongs to different ecosystem attributes. You will see that maybe some of the bulk property might not be changing, but maybe some of the different phytoplankton types or size structure might be changing when total chlorophyll might not be changing. So looking at different indicators is quite useful. I will give you an example now. So just briefly some network of observation. So I've mentioned here different observing systems. So the continuous plankton recorder, so it's based, it was developed at Safos, so on the other side of the Ho. Here, Alistair Hardy Foundation for Ocean Science. So here is an image of a continuous plankton recorder. So this is a device. So it's you get water coming through. So it's attached on a ship. So ship of opportunity. So not necessarily a research cruise. Actually, it was more commercial ship traveling between the UK and the US, for instance, crossing the North Atlantic. And you get water coming through, and it's collected on the silk. And well, okay, so you get information on zooplankton, but also on the phytoplankton. So the phytoplankton cells might break down on the silk, and actually give a certain color of the silk. And based on the color of the silk, then you can get some information on the concentration of phytoplankton in the water. So this time series is very interesting. It started in 1948 about, so it's several decades of observation, I think 6 or 7 decades by now. Atlantic Meridional Transect, so it's one example of research cruises. So PML is very, very strongly involved into these cruises that go across the Atlantic. Maybe some of you have been on these research cruises from the UK. Definitely Bob has been on several of them. I think Dionysios as well has been on a few. And for some time they were running during spring, summer and autumn. I think in more recent year, maybe more autumn season. Visible spectral radiometry, so a range of sensors. And for instance, here I put BioArgo. So these are equipped of sensors that can measure fluorescence. And we can also retrieve some information on some of these indicators. So for instance, what I classified under glider would also apply to BioArgo floats. Okay, so trying to get information from different observing systems and combine this information with satellite observations, and also combine information from indicators from different ecosystem attributes. So what can we get? So here is okay, Africa, South America, and we see 3 biogeochemical provinces, so Longears provinces, North and South Atlantic gyres. and the tropical Atlantic region here in the middle. Actually, just that part. And so here we have the cruise track from the AMT. And we see, so it was a simple calculation of trends over about 1 decade of data. So using just one satellite, so CWIF in that case, and the in situ measurements. And okay, we found no significant changes in chlorophyll concentration in these different provinces. However, using in situ observations of pigments, we actually found some significant changes in the phytoplankton size fraction in the different regions. So decrease in the microphytoplankton and an increase in the nano and pico phytoplankton. So what does it tell you is that even if you don't see changes in the total chlorophyll concentration, actually the size fraction might be changing. So this is quite interesting information, and we could only see this because we used different stream of observation and we looked at different indicators. Another example where, so these are examples based on my studies. Of course, there exist many in the literature, and it's not at all exclusive. It's just examples that I know. You can find many others. Probably you have developed some. So here is looking at PhySat. So I introduced it just earlier. So this method that allows you to identify different phytoplankton functional groups. And we looked for the North Atlantic at periods when we had a positive North Atlantic oscillation in red and negative North Atlantic oscillations. And so this is from satellite observation, and this is from in situ CPR. So CPR also sometimes they can detect for the larger phytoplankton cells, they can have some information. And so we found similar patterns in terms of anomalies of diatom biomass and anomalies of dominant diatom frequencies from PhySat. So this is an example where okay, we use different stream of observation. And a final case study is looking at variations in phenology in the North Atlantic. So using a continuous plankton recorder and satellite observation. So here are the tracks from the continuous plankton recorder. So over 5 decades. So these are all based on ship of opportunity. So it means it's a voluntary process. The captain of the ship agrees to carry this continuous plankton recorder and measure the, let the water pass through the silk and send it back to Saphos for analysis. And so you see, so what can be done is just based on the color of the silk. So you have 4 categories. So it's all done visually. The person, so it's a long training process in order to for the measurements to be comparable. So it's and to avoid too much subjectivity in the measurement. So they have 4 categories. So it's either it's not green at all. It's white. It's very pale green. It's pale green or green. So these are the 4 categories. And actually, a colleague, Dionysios, here at PML, has been looking to, from these 4 categories, to convert this into, to translate it into a concentration of chlorophyll. So he has a paper on this, and using his equation, then we can have a phytoplankton color index expressed in milligrams of chlorophyll per cubic meter. And you see that then it's based, it's displayed on a grid. So this is a grid of the phytoplankton continuous plankton recorder. It's boxes, larger boxes like this. And then you can have an estimation, an evaluation of the chlorophyll concentration. And these are time series, so it's a different way to look at it. So here you start 1960, and you go to 2000 up to about 2010. And here is a time during the year. So it's actually expressed in weeks. So this is a week number 10, week number 20, week number 30, and week number 40 during the year. And you see, so this is about the time of the spring bloom. so maybe around May, April. And here is from the satellite observations. So I'm not sure if Shubha mentioned CZCS. I don't think we've mentioned it at all so far. CZCS? Not really. So it was a... Okay. Yeah. So CZCS is a coastal zone color scanner. It was the first ocean color sensor that had been launched. So it was a very exploratory mission. So this sensor was not switched on all the time, actually, not collecting data all the time. It was mostly switched on when it was passing near the coast, and especially in the northern hemisphere. And it was operational between 1978 till 1986. So we have about 8 years, 7 years of observations. But it didn't have the same wave bands as other satellites such as SeaWave. So it's actually not so straightforward to compare absolute chlorophyll values from CZCS with absolute chlorophyll values from other sensors such as SeaWave. So actually what we proposed was to compare changes in phenology, so which are based on relative patterns. So they could be more directly compared. And we also compared this with CPR observations. And so it's for two areas in the North Atlantic. Area 1 is on the top and area 2 is at the bottom. And so every year we estimated the timing, so when it starts here and the termination. And so this is a growing period at every year based on the continuous plankton recorder. And in blue, it's same estimates, but based on the satellite observations. And so you see here is for CZCS, and here is one for CZCS. So there was a lot of gaps, time series that are not continuous, so it's not always straightforward to estimate phenology. But for some years, we succeeded. And so we see that in area 2, actually, we get relatively good agreement between the satellite and the continuous plankton recorder in situ observation. Well, here is actually very difficult to make a direct comparison. And with CZCS, okay, we get very few points. But so this is just an example, and it's also shows you the challenges to make comparison, even when you use different indicators, relative indicators, to compare different stream of observations. Okay. So I've mentioned a few times about missing data in ocean color time series. So missing data at higher latitude would be due to a high solar zenith angle. In some regions, so we don't see here, but for the Arabian Sea and another region of the ocean, when you would get a lot of storms or very, very high temperatures, and you would also get a lot of missing data, so you get gaps in the time series. And having gaps in the time series can lead to errors in your estimation of the ocean color indicators. So phenology, definitely, you might miss a timing, but also chlorophyll and other variables. And so I did an interesting study where actually maybe it was needed. So the CZCS sensor shown here, so this record is quite far back in times, and of course when you're trying to look at influence of climate, whether it's climate variability or climate change, you would like to have a time series from as far back as possible. So here it shows you the availability of observations from the CZCS sensor during its lifetime. And so you see that really along the coast you had a higher number of observations. So total number of observations. In the southern hemisphere, very, very few observations. And if you compare with a satellite sensor, so the next one, very strong sensor, so SeaWave, over its lifetime, but you see you get much, much higher availability of observation, higher density of observations. So in order to estimate the influence of all these gaps, all this missing data, and whether actually we could retrieve any information that was useful from this satellite. I did a simple study. So where I'd taken the CWIF time series as a reference time series for satellite observations, and from this gap-free time series, gap-free, containing the gaps that you would expect from a satellite ocean color sensor, I applied the gaps of the CZCS. So I took the CWIF time series as it is, and I took it again and I applied the gaps. I applied like the CZCS gaps as a mask. And so I had a gap-free and a non-gap-free time series, both from CWIF. So I estimated climatologies, and I estimated different indicators. So chlorophyll concentration annual mean, the median value. I looked at phenological indicators, and so I get an indicator reference and an indicator from the time series with gaps. So I repeated, and then I compute the difference between the indicator from the time series with gaps minus the reference. And I shuffled around the different years of CWIF, and I always apply the same mask from the CZCS. And I repeated this procedure 25 times and generated 25 unique gap-free time series. So yeah, I had 10 years and I only kept 6 years. So I had always a climatology estimated based on 6 years of observations. And to estimate the uncertainty, I looked at precision. So here is how far you are from the actual true value of your observation due to random noise or just due to gaps in the data, and also looking at accuracy. So actually this is how far you are from the true value. So it's like estimating your bias, and estimated the precision you're using root mean square error on this difference, and estimated the bias simply. And this is some of the results. So for peak chlorophyll concentration, so the maximum amplitude in the chlorophyll, median chlorophyll and mean chlorophyll. So it's annual mean and annual median. So based on the climatology, and so this is looking at the bias, and so we see that as soon as we reach about 60% of data coverage or data availability from what you would get normally, then your actually bias is very, very small. I mean, it's only a few percent. Of course, if you get time series with 80% or 90% gap, you get much higher bias. Same for the peak chlorophyll concentration. So actually looking at maximum values, you get much larger bias. We are more around the 20% bias. And this is so a measure of your precision, so root mean square error. And so this is always in the positive. And again, if you're around 60%, of availability of data from the CZCS, well, okay, then you're around maybe less than 10% error in precision. And for the phenology indicators, so here is the bias in relative percent for the duration, and root mean square error for the duration. And at the bottom, it's expressed in days. It's by how many days you are likely to miss the timing of peak or be off. So it's actually quite large. I would say around 60% of missing data, then you get a bias of maybe about 20 days in some cases. And an actual precision is actually quite low. So here is a median in the middle. So you'd get maybe around 40% or 40 days in that case, and 40% in terms of duration. And so at the end, what we got was, I didn't show the final figure, but we get regions. We are able to estimate in which part of the ocean we are able to estimate with an accuracy and a precision of so many percent. Let's say we put a threshold of 15% accuracies, and we know exactly which region we can have estimates of duration, of chlorophyll concentration, or other phenological variables. So then we know which region we can use the information from the CZCS. So this was the idea. Okay, and almost last slide. I think I'm on time, or maybe I'm way past. I didn't check, but nobody complained. I suppose. Okay. So very briefly, just one slide, 2 slides on the use of just one example on the use actually of phenological indicators for model evaluation. So comparing satellite observation and ecosystem model outputs. So you can have a chlorophyll concentration from ecosystem models. Of course, quite classically, maybe you would look at changes in amplitude. So this could be your observation, and this could be your model. So maybe your model might be overestimating chlorophyll concentration or underestimating in other cases. You might look at median chlorophyll concentration to do a comparison between the model and the satellite observation. But you can also look at changes in timing, and so phase shift. Simply, here is an example: satellite observation, timing of peak. So again, the color scale, it tells you which month or which week of the year the phytoplankton bloom peaked. And you can do the same analysis in the model, and then simply you can compare the two. Here is satellite minus model. You might want to do actually the opposite, more the model versus the reference. But here it shows you that in regions that are in red, is simply that the timing was earlier in the satellite compared to the model, and in region where you have blue color, the timing was later in the satellite compared to the model. So these actually have quite big implication, because if you have in your model, so your model might be accurate in terms of amplitude, but if you compare at this time the chlorophyll concentration, you might think, my model might be underestimating chlorophyll, while actually it's simply that your seasonal cycle is just shifted by maybe just a few weeks. So it's actually an interesting metrics to use for model evaluation. Okay, just one slide on conclusions. Yeah, okay, so I'll read it. Remotely sensed data are useful for the construction of time series, but you need to pay close attention to quality control, and also, of course, as I showed, gaps in your time series. Ocean color time series provide a cost-effective basis for development of ecological indicators, average at appropriate time and space scales. Just 2 environmental variables, so chlorophyll and SST, can provide already a very rich set of ecological indicators that can be derived from these 2 variables. A subset of the available indicators can be combined and aggregated to make an ecosystem index of ecosystem health. And so this ecosystem health index can also be complementary to other measures of ecosystem health. And just from the last slide, for the purpose of comparing models with observation, so phenology is a very interesting metric to look at. Okay, thank you very much.